So now we will be drawing the final structure after secondary growth. So in that, firstly the boundaries, the outermost layer which will be epidermis, few layers of cork, cork cambium, secondary cortex, phloem, secondary phloem, then there will be the 2 or 3 layers of vascular cambium, primary cambium. Then we have secondary xylem, it should be double than secondary phloem, and in the ground tissue. Now this is called ground tissue. In this region we will have few elements, large elements of xylem. Okay. We are not going to show primary xylem and all, just this much part. So the outermost layer epidermis, which gets ruptured at few places. So you can show some gaps. Then we have the cork cells. Leave the areas below ruptured part to show lenticels. Perfectly aligned cells, this part you already know. So I will complete a small portion only. So this is cork. Similarly on the other sides also you can draw, and in this region we have lenticels. So loose parenchymatous cells. Then there is present cork cambium. So cork cambium exactly aligned below lenticel also 2 layers of rectangular cells which forms cork cambium. Okay, now the next part is secondary cortex. The parenchymatous cells leave some space towards secondary phloem, because where both merge we have abundant latex canals. Okay. In this region also you can show abundant latex canals. You can draw large polygonal cells which will mark latex canals. And I am thickening those cells so that they are differentiated. But the majority of the latex vessels are found close to secondary phloem. Okay, so you can draw many towards this zone. Rest of these will be the cortex cells. Now next to it is secondary phloem with these latex canals. Draw large sieve tubes. They may have straight or oblique sieve plates. Now rest of the phloem elements should be very small. You can draw medullary rays also. That is the interconnections between secondary tissues. So, smallest cells of phloem, polygonal cells, complete this area. Then we have 2 to 3 layers. You can show more because in the vascular cambium many layers are seen, but here we are going to draw only 2 layers. So, 2 layers of vascular cambium. Then we have xylem. Now, here I used red color, so I am going to change the color in this case. I am using slightly pinkish color, okay, in order to differentiate it from cambium. So, the vascular region will have more of the parenchyma, very few vessels. So, the vessel areas you can mark specifically, the large areas. Show just few vessel elements. In this way you will complete secondary xylem and make all the cells double walled. Parenchyma cells are also present in between, but we are not going to differentiate that, and marking all cells with double wall. Okay, this way you can complete this part. Now, inner to this, which is the anomalous part, we will have few xylem elements which got separated from secondary xylem. They may be present singly, they may be present in groups. So, I am showing here 3 elements, double walled. Now, see this part carefully. Around this we will show the cambium. Okay. These are secondary cambia which develop around the separated vascular elements in ground tissue. The tissue surrounding it is ground tissue, which is having abundant storage parenchyma. So, you can show 2 to 3 layers of these secondary cambia around vessel elements. One cell over another, and outside this just very few phloem elements are cut, which include sieve tubes and few latex cells. We are not differentiating both the cells, we will be forming just one layer. Green color I have already used for phloem. Okay, so you can have an idea that this is the phloem tissue, and adjoining this there will be few latex canals. Here also we show latex canals, so in this also you can make few latex canals adjoining phloem. Now rest of the part is all storage parenchyma. So in this region again you can show polygonal cells or circular and oval cells. So fill the space, mark some line for the initial one. And all storage parenchyma, they have food stored in the form of starch. I'll show you the real cross section and you can see yourself. They are laden with food completely, all the cells they are filled, and in order to stop conduction in this region, xylem vessels they form tyloses. Okay, tyloses, the outgrowths of parenchyma cells, xylem parenchyma in the lumen of vessels and tracheids, so that they stop conducting. Okay, so these are latex canals. Here also. So this is the structure, and towards outside we have cuticle. So the labeling part: cuticle, epidermis, cork, cork cambium, secondary cortex. These three comprise periderm. The cork cambium arises from pericycle. Then we have secondary phloem, latex canals, vascular cambium, or we call it primary cambium. Secondary xylem. Again I am telling you very few vessels are present. Ground tissue, secondary xylem elements which get separated from this tissue, secondary cambia. These will cut xylem towards inner side also. Okay, we haven't shown, but small xylem elements, they are cut towards the vessel. Latex canals. So this is a part of, it's not complete TS. So a part of TS of mature root of sweet potato, showing secondary growth. Okay, the diagram in your book is not correct, so you need to follow this diagram. So in this case, finally we have observed that abundant, it's a storage route, so abundant storage parenchyma develops, and basically there is two type of parenchyma. What are those two types? One, which is the normal bundle parenchyma, means the parenchyma developed along with the secondary xylem and secondary phloem from this vascular cambium. So it develops from primary cambium. And the second type of parenchyma is the storage parenchyma, which is interstitial. Interstitial means present in between the secondary elements. This is called the storage parenchyma. So that is all about the secondary growth or the secondary structure of sweet potato root.